I have a question. So whenever we complete the course eval, do you want us to take a screenshot or something of it?
Yeah, it's like the last screen. It doesn't say anything about the class. It just says you have completed it or whatever.
Send it to you?
That's, you'll post that on the actual assignment,
Okay.
like a submission in the assignment, yeah.
For what?
For the course eval stuff.
Oh, fuck, I already exited out of that.
Oh, you can go back into it and it'll just be like you completed this already and just
Yeah.
take a picture of that. It's fine. If you already did, just take a picture, whatever it has. Like I trust. Like it tells the system like how many people have done it, so. Okay. Well, I had opened this already, but apparently.
I'm sorry.
It should be in, like, it'll be like other extra credit or something.
It's just at the very bottom of the assignment.
Yeah, and I'll be posting an announcement about it. I just haven't gotten to it yet. Which is for also me saying that like today, my plan for today is to grade the lab report stuff. I haven't done it yet. I did not have time for it this weekend.
I do not blame you.
Yeah.
You have like how many students and all of them had to do that?
Yeah, 140 of them or something.
I won't lie, every time you graded something over the weekend, I was about, like, I'm gonna throw up, because I don't know how I did on that.
I like graded 2 of them and then I was like, that's all I can do. That was very busy.
Oh, I just.
My husband asked me to get his tires replaced in his car. Guys, he replaces the air in his tires every day.
What?
Every day they go to low pressure, like, and you can see cracks. They're just old, right?
Seeing the price of tires every day?
Right, well, he needs
Hold on, there's
tires.
a place in Moore that does them for really cheap. I got all 4 of mine for under $700.
Really?
Yeah.
Oh, that-
700. I got a little
Jesus.
more.
I buy like pre, ones
Like,
that
I
are
got
so
really,
great
really nice
condition,
tires.
and I just do that, and there's one right off the bat.
I'm fine with it. And it's like, I don't mind, but I would like,
For like 300.
yeah, I want to get somewhat decent ones because he decided now suddenly he wants to get a new car, which we've paid off both of our cars. So of course now naturally one of us wants to get a new car. So he's had his car for over 5 years. I think it's been like 6 or 7 actually. So it's fine. You know, I think every 5 years would be fine. And now he's made it longer. So he has a Honda Civic, so it's going to have great resale. I'm not worried about it. So it'll trade in well. But he's jealous of my Jeep. So now he wants to get,
Oh, gosh.
he wants to get a Wrangler, which I'm jealous of. Because the whole reason I didn't get a Wrangler is because it's so expensive.
What
So I drive,
What is he doing?
oh, he does his Honda Civic. So
I will say, it
But-
sounds like usually a lot better price.
then trade-ins,
Yeah.
yeah. But I mean, I'm not too worried about it. Anyways, so he wants to replace well, first of all, he needs to replace the tires, but he wants to replace them so they're somewhat nice. And we're gonna get the full detail of the car and everything before we go have somebody look at it and appraise it and all of that, so. But anyways, I was supposed to take this car, the whole point I was bringing this up, supposed to take this stupid car to go get it, replace the tires. And I locked my keys in the house on my way out the door. Like I had like locked the door behind me because it's like a handle or whatever. And I locked it behind me and shut it with the keys in the house. And I'd also set the alarm to the house. So I'm like, I guess we live in the garage now, like.
We have the locks that have the keypads on them for that exact reason.
Yeah, that's sort
Like
of,
kept locking us out of the house.
yeah, we're gonna have to get one. We've been looking at them, but they're not cheap.
Definitely feel your struggle. This morning, so my brother lives with me, so there's 4 cars in front of my tiny house. And I had put the trash can out so we could all pull out, so I guess they didn't think they could pull out, so they moved it behind my yard. I didn't realize that, and I flew it over.
Uh-huh.
Because I had to go and pick up trash.
Yeah, you're better than me though. What I did one time, my husband's side of the garage was filled with boxes for a long time when we moved into our house. So he would park behind it, like it was 2 spaces in the garage. I had my side cleared out. I cleared my side out because I wasn't doing anything. So I cleared my side and had my spot that I'd park in. And for whatever reason, he always parked behind the trash. But one day he just parked behind my car, and it was late at night, and I forgot I'd left my space heater on in my office. So I was like, oh shit, I need to go turn my space heater off. I can't leave that because it doesn't have like a really cheap one, right? So I like just go and get in my car and start it, and it was a cold day. That's why the space heater was on, and it fogged up my rearview mirror.
Oh, no.
And he never parked there, and he was parked so close, I couldn't see him in my side mirrors. I just backed right into his Honda Civic with my SUV. It did nothing in my car. But I audibly heard the crunch in the front of his car. So there's a big crack. It's just a superficial crack, but it's just one of those things where I was just like, I like pulled forward in my car. I don't know how to approach this. Should I be mad at him or me? Like, I don't know.
I would still find a way to be mad at him.
Of course I was mad at him. Yeah, I'm not sure that was fair.
You never parked there. What's your problem?
Yeah, that's what I said to him. I was like, you never, and I fogged up my mirror legitimately. My car didn't even see it. Like usually beeps when there's stuff behind it.
I'm so happy for you guys, because I'm having the worst time of my life with Carl.
All right, guys, go ahead and sign in and we'll get started with our review. I'll do the best I can for you guys. So in case you weren't paying attention, the study sheet that I provided for you guys, we have the lab study guide, which is pretty good for filling out and just making sure you're familiar with the info. We have the study sheet that like lists, there's two different versions, the table that lists like the tests that we did and the descriptions of them, as well as the outline with all the actual pictures associated with it, which is a million pages long. But I've separated them because of that. So there's that. There's also the, that's just for the tubes. There's also the selective and differential media guide that is available on there. And there's also, well, I guess the study sheet. And then the stuff that we learned from last time.
Super helpful.
I love these, please never stop doing these.
Yeah, never.
We're so trying. We tried. That's what took me so long last night. That's why I didn't get to the grading
Helped me
things.
so much.
They're so fun, and we can just continue to redo them, and that helps too.
I work on this, I work on the study sheets, all that stuff. I hope it helps you guys. Luckily, it means that less work for me next semester, but
The repetition is what helps so much for me, and going over it and over and over.
Yeah.
Like that.
I feel like this next test too, because you guys kind of now know what to expect. Usually the second lab test goes better, because people know it.
Wednesday in here.
Wednesday in
Well,
lecture,
I have my presentation.
it's just gonna be review, or yeah, it's gonna be presentations, that's right.
Presentation.
Just presentations. I'm gonna tell you guys what we're gonna do on Wednesday, since, I know some people left already, but that's okay. What we're gonna do on Wednesday is the presentations, whatever four, I think we have 4 of them that are left over that are like carryovers. And whenever we are done, directly after that, we're just gonna go take the lab test. I don't wanna wait till 11:30. Why don't we just knock it out right away and get it taken care of? And then you guys don't have to worry about it anymore. So that's what we'll do. We'll do presentations and then straight up into the lab exam. Of course, if people show up at 11:30, you know, they take it, but yeah, so we'll do it that way.
And then what day is our lecture final? Like what will we be doing in lecture leading up to?
Right, so today was our last lecture, then we'll have the remaining presentations on Wednesday. Monday, we'll have a review. Wednesday, we'll have a review. All again, optional. There will be no points tied to it at all. And I'm just going based on what you guys said you wanted to do in that quiz thing that I posted on Kahoot. So it seems like the consensus is for both of those days to do some sort of Kahoot exercises. That's fine. I'm fine with that. I'll put some Kahoot type stuff together. I might have at least one of those days be a competition day, or we'll have like, you know, you pick a group that you want to be in, and then it'll be group against group kind of competition. I usually have better involvement that way. I mean, we have fun enough with just individual, but I feel like the group versus group is a lot more fun. So we'll see if we can't knock that out. But yeah, I don't know how I'm going to divide it yet. Probably the first day will be unit 1 and 2, and the second day will be unit 3 and 4. That makes sense to me, right? So we'll probably do it that way. It'll be in here. Yeah, for sure. So that'll be next Monday and Wednesday. So easy enough, right? No points associated with that. So if you don't want to come, it's okay. Also, I'll probably record it and Zoom it at the same time if I remember. That's the hard part. Then the Monday after that, of course, I don't know how, will be the exam. I don't know how that's happening.
Yeah.
But it's happening, okay. Huh? Yes, I'll for sure have, like in class, we'll play them and record them and Zoom them and everything. And then afterwards, I'll post the actual links online. Just because I usually try to fine tune them up until the last minute so that we have something to work with. And I know most people like work ahead or anything. To get advantage. Okay, so let's see. So let's start and go ahead and review for the lab. I mean, we really are basically done. I like that. That's great. And I'm not complaining. All right, so you guys remember the final, it's gonna be the same format as the midterm. It's pretty much gonna be the same thing, just new stuff added in. So 24 to 26, I haven't completely settled on what questions I want to go with yet, so somewhere in there, but about the same layout, 50 questions, all multiple choice. 1 minute per station, just like last time. We'll go through the timer first, and then whatever time is left, you'll have plenty of time to go and visit afterwards. I mean, it really is gonna be the exact same thing as last time. Just add in new info, right? So be sure you bring your number 2 pencils. I'm gonna try to order some new number 2 pencils. Hopefully we'll be here in time from Amazon, but you know how that goes. So let's start out with our culturing basics. What is most commonly used to inoculate cultures? Easy enough. This is the kind of question I think that girl was looking for. Yeah.
You have Amazon Prime?
Yeah.
Can you use your like, okay, say, you know?
I can claim on tax returns. So that's what I've been doing anyways. So I'm getting a much bigger tax return now. Luckily, I work for the state technically. Yeah, I use my regular one, but it's tax deductible, so it's not a big deal. All right, so loop, absolutely. Yeah. Yeah. All righty. In which quadrant of a street plate would you expect to find the most single isolated colonies? So look for the isolated ones. It's one of those days I'm so thirsty, but also like peeing it out immediately. So good job. That's gonna be quadrant 4. Good, good. All right. We already know all about aseptic technique. Remember, the whole point of it is to prevent contamination one way or the other. It doesn't necessarily have to deal with pathogens, but pathogens and all contamination. So lab safety, remember, what do we wear in lab? The lab coat, yes, and closed-toed shoes, preferably if we were in a lab. Types of media, this is just going to be talking about like broth versus plate versus the slant and all of that, butts, all that stuff, right? And then the different ways to inoculate the media. So whether we're talking about the loop, the needle, the loop and the needle, if we're talking about TSIA, remember how that has both going on. So, all right, stains. Any stain we've done up to now, right? You will not see any stains that we did not have on the previous test because I only have limited stain slides.
Oh, no.
So, right, so oh darn. So, all right, what is the first step in the gram stain? Right, so the first stain is going to be crystal violet. Remember, it's crystal violet, iodine, decolorize with the alcohol to remove what didn't set, and then counterstain with safranin. Primary stain is going to be crystal violet. Primary and first are the same thing, literally. So what color do properly stained gram-negative bacteria stain in the gram stain procedure? You're gram-negative. All right, so this one's going to be red, negative, red, commonly negative is associated with red as a color. Remember, positive is purple. Our 2 choices are red or purple. I'm not ever going to throw in anything else to trip you up. It's only ever going to be red or purple. So don't think pink is going to slide in there because it's not. What is the name of this staining procedure? It's one pure culture. This is not a mixture of bacteria. I will say this, if there ever is a stain that I put in a microscope on the test that's coming up, that is a mixture, I'll say so in the question. So just always assume otherwise that it's gonna be a pure culture, okay? So that's what basically tells us that's the endospore stain, yeah? All right, which of the following bacteria will be acid fast positive? Uh-oh, acid fast. Let's see if you can tell which word that looks like the other one. All right, our answer here is going to be Mycobacterium flea.
You should be probably gonna get out soon. Would you explain why?
Why it's Mycobacterium flea?
Yeah.
Because it's mycobacterium, only mycobacterium, yes. So mycoplasma, they have no cell wall. So they have their own thing going on, but yeah. All right, so after the slide has been heat fixed, when do we use heat again in the acid fast stain procedure? In acid fast. In acid fast. Think about it. What do we do in the acid fast stain? The acid fast stain, we put quinones, carbofusion on there, wait 5 minutes, rinse it with acid alcohol, rinse it with water or in between, right? And then counter stain with methylene blue. That's it. There's no heat in that one. Only the endospore stain has heat. So that's, I think, the next question, just FYI, I'm just saying. Oh, not yet. Only endospore has heat. Yes. What color is negative acid fast stain? So when I'm talking about heat in the procedure itself for the staining is what it's asking about. So, what color is negative for acid fast stain? So if mycobacterium are acid fast, they would be one color, but what are the others going to be? So this is blue. So the carbofusion will stain acid fast bacteria like Mycobacterium fuchsia, but we counter stain with methylene blue. That'll stain everything that is not acid fast blue. Y'all doing pretty good otherwise though. All right, what is the name of the primary stain used in the capsule staining procedure? So I want to point out this picture to you guys to remind you of what the capsule stain looks like. We did very poorly on this on the last exam. Everybody thought I was trying to trick them because it had purple and pink in it. They thought that must be some sort of weird mixture of gram stain with a weird background or something. I don't know what. But that negative stain that creates that background, the first stain we use is Congo red. We put medieval stain on top of it. It turns it purple in the background, but it leaves the cells that this nice bright pink color from the medieval stain. But yeah, so the primary stain is Congo red. Don't forget what that looks like. You're gonna see a capsule stain on the exam. So just FYI, I'm just reminding you so that you know what to look for. That's why I put that picture up here so it was identifiable. All right, in which staining procedure do we use steam? So we said this one just a second ago, but I'll say this for the back on the capsule stain side. For the capsule stain, remember what it looks like to be positive and what it looks like to be negative. So in the capsule stain, if you have a capsule, you have that halo, the white halo around you. So keep that in mind. That's a capsule. It doesn't stain. Well, yeah, endospore for steam. That's the only one that we use heat in the middle of the staining procedure. So know the procedures. I don't care about how many minutes the stains go on there, right? But which stains are used, what order they're used in, how they work. I'm going to ask less questions about the stains than the tubes. So it's going to be mostly the tube stuff. You guys know that expect that's the new stuff. I ain't asking any questions about a eukaryote. There'll be like one station about the microscope and it'll be clearer than the last few ones. Yeah, it won't be that one. So look out. Now that we know that what that one is, nobody's gonna do it. All right. All right, so differential and selective media. These are all those different plates that we had all at once. So what type of media is this one? So this is one I couldn't find you guys a good example of. You guys had one, I think, in your class, but this is what it should have looked like when we had this happen. All right, so this is EMB. EMB, that was that metallic green I was telling you guys about. That meant you are aggressively utilizing lactose. So what is the selective agent in EMB? Selective. Remember there's selective and differential media. Is this, what's the selective? All right. Our selective agent in this media is the EMB itself, eosin methylene blue.
Yeah, I did too. Like, too easy.
All right, what media is this one? See if you guys know it.
So much fun in this class.
All right, great job, guys. This is in fact McConkey auger. All right, there could be more than one in this. Is this media selective or differential? Could be more than one. Good job. Really good job. Yes, so this is both selective and differential. All right, which media is this one?
I think that's one.
All right, good job. This is mannitol salts auger. We're going to talk about them a little bit at the end. What is the pH indicator used in this medium?
She's like, yeah.
I'm just thinking about that on the way.
I have no idea in my car, maybe it's 2 hours.
All right, perfect. That is going to be phenol red. That's why we have that orange to yellow color change. That yellow is from the acid production from fermentation of mannitol. So it makes it into acid. Okay, for the differential and selective media, there's a lot going on in each of these plates, actually. Which one is selective versus differential? Maybe they're both, maybe they're neither, right? Remember we learned about even talking about nutrient auger. And nutrient auger, how it's not selective, it's not differential, it's just nutrient auger. So think about that. Which ones are one or the other or both? What the selective agents are and why they're selective or how they're differential, what the color changes are. Be aware if there are any pH indicators, what they are and what the color changes mean. And don't forget staphylococcus media, blood auger, went into 3 types of hemolysis: alpha, beta, and gamma. Alpha is greening, beta is clearing, gamma is growing, but not any visible change. So what I did want to say is, we know that EMB and maybe you don't know for sure, but EMB and McConkey both have evidence of color change of the actual growth itself on the plate, right? If it uses lactose. If you use lactose traditionally, you're going to turn pink. If you're on EMB and you aggressively use lactose, you do that metallic green, like we just saw. So that's aggressive lactose use. So if I were to ask you guys a question on the test, which of the following media no, actually, let's say I give you a plate that looked just like the one we saw. Okay, dark with the green on it. And I said to you, which of the following sugars does this test for fermentation of? Then you should know that it's lactose. But I also have a tendency to give questions worded in a tricky way on this particular exam, which is to say, does this organism ferment glucose? So if I ask that, you better not let that trip you up. You're like, okay, it's got growth, it's pink, so yes, it is fermenting. No, no, no. We do not test for glucose fermentation on EMB or on McConkey, only lactose. That's a common way that I'll ask the question on some of these. So just keep on your toes. Know your tests well enough, and that won't be a trick to you. All right, that's a better way of wording it. So moving on to the biochemical test, the newer stuff, okay? We have the big old handout for these ones. So if you have that available, this is the time for it. So which test is this one? Wow. So that's the citrate, the Simmons citrate auger. Citrate and malonate are the green ones. They're the only two that are green. They'll turn blue. All right. Which of the tubes is positive in that picture? All right, great. So that's going to be B. I kind of just said it, so it's okay. Yeah, but it turns blue. All righty. But what is happening to the pH in tube B? Now, it's turning blue, but what does that mean for the pH? What's going on there? Hopefully, I actually pay attention to changing the answer to this one. All right, so the pH went up. So there's alkaline production. So the byproduct here is alkaline, it's basic. When we break down the citrate, causes the pH to go up. So the higher the number, the more alkaline. That's what changed it. So do you guys remember what the pH indicator is for that one? Bromothymol blue, right? Another thing that I like to trip people up on the test with, bromophenol blue. Don't fall for it, okay? Bromothymol blue is the correct word, not bromophenol blue. People are easily tricked by that one. I like it. It makes me happy. So citrate, how is it inoculated? That's a slant, right? Just a slant, nothing else on there. How is it red? That's going to be that green to blue color change. What colors are produced? Blue is the only change, but it's green if it's original. What do the colors mean? And what is the pH indicator? We kind of already went over that. So moving on, what test is this one next? Ah, good old friend. Okay. Y'all did not do good on this one. So this is phenol red carbohydrate fermentation. The reason that you know that it is, number one, it's an orange, so it's phenol red. So we got that part, right? The other favorite was urease. It also has phenol red. So at least we're in the right category here. But that little upside down tube is what gives it away. That's what I don't want to call it yet, because I think it's a question. But you guys know it captures the gas and the reaction. All right. What is the name of the upside down tube in the inside of that assay tube? What is that upside down tube called? My stomach is growling.
I truly don't know.
Oh.
You know that.
Yep, that's the Durham tube. Good job. Durham tube. That'll definitely be a question on the test, just so you know. It's an easy enough one, yeah. What are the two products of fermentation detected by this assay? There's two possible things from fermentation that you could see, you know, in this. All right, so it's definitely gas. Yes, we know that because the Durham tube, right? And acid is the other one. It's a byproduct of fermentation. I should have put alcohol. I'm really throwing you guys off. All righty. Which test is this one? So the next ones are going to be kind of like this. There'll be a lot of choices till we get through all of them.
I like everything. But how's it on my phone?
All right, pretty good. This is sim, correct. What does the I stand for?
Y'all answered before I could even read. No, we're
Perfect. You guys did great. That's indole.
Yeah.
How is indole detected? What do we add to the tube to see if indole is there? Looking at this figure, could you tell me which one is positive for indole production? Yep, it's going to be the second one. So this one is Kovacs reagent. So how do you detect H2S or hydrogen sulfide in sim, sulfur indole motility? How do you detect H2S? Yeah, so it's going to be a black precipitate. That's indicative of sulfur reduction. How is motility detected? We've got 3 tests. We talked about the sulfur and the indole. Now, how is the motility detected? Yes, it's going to be growth away from the stab line. The best way I can describe it, you guys have seen some of the tubes where there'll be a stab line. There's like some little guys, like fingers off of it. That's not what I'm talking about, okay? That's usually just mess from the actual stabbing procedure. If you look at the pictures that are like in printout, I feel like that clears that up pretty well. You can see the line nice and clear. That's going to be negative for motility. If it is like diffuse out in the media, like a cloudy throughout the media or spread out, stuff like that, then that is going to be growth away from the stab line. So diffusion away from the stab line is probably a better way to word it, but just so you guys know. So how we inoculate it, it's a butt, so we stab it with a needle. Interpretation of the results and the changes, we kind of went over those with this one already, and the addition of reagents, that was Kovacs. So which test is this one? Right, so that one's litmus milk. Good job, guys. Everybody knew that one. All right, in litmus milk, what color is associated with an increase in the pH? Increased pH. As you guys are looking through your choices, if you can tell me what the other ones are doing, at least keep them in your mind. Yes, litmus milk, increase in pH, that's alkaline, so that's gonna be blue. White is reduction, pink is acid, lavender is the original color, so no change. In litmus milk, white is, so you guys are paying attention. You're gonna feel silly whenever you didn't remember. Yeah, it's going to be reduction. So what you guys will have on the test, I won't just have like a question about a tube and might be asking you about it. You'll actually have the tube there. So it will be like two tubes, and one will be uninoculated, the other one will be you know, the test one, and they'll be clearly labeled, and so you'll be asked questions about them. So you actually have tubes to interact, like a white tube, as opposed to me just saying, what if it's white though? There might be like one question about what if it's whatever color. Some of that stuff I just couldn't get it to grow, right? So litmus milk, the different colors, digestion. Remember, that's gonna be clearing of it. It's gonna look like plasma. It's gonna become translucent. How is gas detected in this one? You can, and you will not see it on the test, but you would be able to see it like in the curds, like air trapped in there. And then what is the sugar in milk? Hint, hint, lactose. There's gonna be probably be a question on the test about which of the following test for lactose use or something like that. That's why I keep bringing it up, okay? So which test is this one? See, it's plate this time. Yeah, I know. I can see how you would get the blood auger if it was like a really, really dark blood auger. Blood auger really is pretty red though. So that was starch hydrolysis. So it's where we take the plain old starch plate and then put something on it to turn it dark color. So which organism pictured is positive for starch hydrolysis? Which one was able to hydrolyze starch? All right, good job. Let's do the one on the left that had the big old halo. What is added to detect starch hydrolysis? What creates that dark color? Because the plate is not that color originally. All right, great. That's going to be iodine interacting with the starch. That's why we get the halo, because the iodine's not interacting with starch there anymore because the bacteria ate it. So which test is this one? Now notice there is a tube inside of this tube. I just want to point that out. Yeah, this is gonna be nitrate reduction media. Great job. You guys are getting there. What do reagents A and B detect when we add them in nitrate reduction test? A and B are detecting so A and B detect nitrite. We start out with nitrate. We add A and B. If nitrite is there, we see red, and that's it, and we're done and we stop. That meant that we have converted nitrate into nitrite. That is nitrate reduction, which is what we're testing for. What does zinc detect? All right, good job. So we use zinc to test for nitrate late in the testing procedure. So it's gonna be our second step, looking for nitrate. That's why I say it's late. And zinc is your later one. So that's how I remember nitrate is late. We're testing for nitrate with zinc. And if it turns red when you add that zinc to the nitrate, the nitrate is still there. We did not reduce or have any nitrate reduction. So, negative. All right, nitrate reduction assay, know what is detected at each step. So we just went over that one. Red is not always positive in this test, right? So if you get it first, it's positive. If you get it later, it's negative. Don't forget the gas. There is a little tube in there, right? That it's still a germ tube. If it collects gas, it's a germ tube every time. We didn't really get any of that in our class, so we hadn't really gone over it too much, and you're not going to see it on the test. But hey, there could have been gas in it, just saying. Which test is this one? Oh yeah, look at this one. All right. This one is the TSIA. Exactly. Finally came around. All right, there could be more than one here. Which tubes show evidence of H2S production?
I clicked both of those and it only did one, so I'm dumb.
Yeah, so this is going to be both number 5 and number 3, the ones with the black precipitate in them. The same as for the sim, it's the same indicator. So the next one's gonna ask about gas. I want you to not include the black precipitate tubes, not the H2S ones. Which other ones show evidence of gas? I know.
I feel like I'm looking at
In my
All right, this one's gonna be 4 and 4A. Those are the two that had the auger like lifted up out of the tube, kind of.
Oh, okay, cool.
That was all gas underneath them. So that's all we're talking about with TSIA apparently. How is it inoculated? Remember, TSIA is going to be a slant. So you squiggle the slant and you're also going to be stabbing it with a needle in the butt, right? So both of those things. How do we read it? We're going to be looking at the color changes. It doesn't just only turn yellow. It can also turn pink. So yellow or pink or no change technically, right? It also could have H2S production with the black precipitate, or it could have gas production like with lifting out of the auger or bubbles on the auger as well. What do the colors mean? This is phenol red. So yellow means acid. That means we're going to have fermentation of whatever the sugar is. We have pink for alkaline. That's going to be peptone usage, just like we saw with the liquid tubes, right? With the germ tubes. We have the black precipitate and the no change if it's still orange. And if we are talking about the colors, slant over butt, they mean different things, right? So slant yellow is acid, meaning you have fermented lactose and sucrose. If the butt is yellow, then you have fermented glucose. If you have pink slant, then that is going to be peptone catabolism, so breakdown of peptone into ammonia, which is basic. And that's why it's pink. That'll be an aerobic version. If the butt is pink, then it's an anaerobic version. That's what those colors mean. Our pH indicator, I've said it 1,000 times, it's gonna be phenol red. All right, which test is this one? Please tell me this one is. I'm really bad heartburn today. Oh, I'm good. I have some in my office. I'll be all right. Thank you, though. So yes, this is the urease test. Yeah, I highly prefer Mylanta, but Mylanta is not really something you share with people because it's liquid. It's like the.
Oh, I've got the pills.
I like it. I don't know what it is about it. I like the liquid. All right. It acts really fast. I guess that's why I like it. But what test is this one? Looking at that tube. What would, so this is the positive version of this test. What would the negative version of this test look like? Right, pretty much, yeah. So catalase, this is the catalase test. So this is adding hydrogen peroxide. It'll fizz up if it's positive. If it is negative, it just won't. It'll look like water sitting in there, basically. So catalase breaks down hydrogen peroxide into water and oxygen. All right, which test is this one? It's like earwax. All right, great. So this is oxidase test. So remember that oxidase is testing for the presence of cytochrome C oxidase, which is one of the molecules in the electron transport chain for aerobic respiration. So it's dealing with aerobes. That's what that's looking for. You just add that oxidase reagent. You don't need to know what it is beyond that. If it is purple, it's positive. If it isn't, it's negative. All right, other tests that you would want to be familiar with that I didn't show in this review, malinate. Now, you guys remember malinate. I don't have a blue tube. So if I ask you, is this test positive or negative? It's negative. We haven't got any that were positive. So FYI, it will be on the test. It has to have that one on test, but like, I mean, I'm pretty sure I have that, but I might not include it, but probably will. It's just easy one, right? Deaminase, that's the one with that slant that looks normal, but you add those yellow drops to it and the whole slant turned green if it was positive, right? It was very obvious if it did. Then MRVP, that is the one where we had the normal looking tube, split it into the two tinier tubes, added some drops to them. One was out orange and was negative. If it was red, it was positive. The other one, it looked like golden goo on the top. We sat there for 30 minutes, it still looked golden. If it had turned red at the top at all, it would have been positive. So the red is positive for both of those. It's two different tubes in there. Gelatin, that's the butt that we stabbed. And then we incubated it, got it out when it was done incubating, put it in the fridge. And when we took it out, it was still liquid. It meant the bacteria had broken down the gelatin, right? So that was the idea of what that was. The gelatin?
Yeah.
And something satisfying about it, I think. All right, so back to the basics on the bacteria stuff, just a refresher. How would you classify this organism's characteristic arrangement that we see? There's an arrow pointing at what I'm talking about specifically. So there might be several arrangements in here, but if I ask you what its characteristic arrangement was, you know, we're going to say, oh, it's primarily that. So this is clearly strepto. That's the chains. We all did see some diplo in there for sure, but. All right, what is the name and shape of these bacterial cells? I don't need a name. I think it's just the name of the shape. I think it's supposed to. Throwing me. Yeah, it's weird. So the shape. What's the shape of them? That man. It's just a weird wording. But yeah, it's not gonna be caucus, first of all, because caucus, I misspelled it with 3 C's apparently. But yeah, it's a bacillus. It's just a rod shaped cell. Rod is bacillus. Caucus is round. One way you can remember is to just like hit me suddenly. That caucus is the round one. The C's and the O's are all round. So if that helps you remember that, it is caucus. All right, bacterial cells, their shapes. That's really just gonna be bacilli and cocci because I don't have any pictures of anything else, right? No slides. Or anything. The arrangements, I'm not gonna ask you about anything that you're looking at, but it might ask you about it. There might be staphyla. We had staphyla last time, right? That was a pretty obvious one that was grouped. That's the only one I have that's a distinct grouping. So that'll probably be the same slide. Just saying. Don't get don't get shapes and arrangements confused. A lot of people think that strepto is the shape of a thing or, cocci is an arrangement. That's not, don't get them confused. Cell shape, bacilli and strep, and I'm already doing it. Shape, bacilli and cocci, arrangements, staphylo, strepto, palisade, diplo, that sort of stuff. But you're really only going to see staphylo or not. I don't know what to tell you. So yeah, remember that simple stains are a thing. That was a thing that a lot of people who wanted to look at their samples one last time when they're unknown kept forgetting. I had several people in random classes here or there that were going to do gram stains to look at their shapes one last time. I was like, you can just do the simple stain. And they were like, what was that? The very first one that we did. And remember, that's easy enough. It's going to be, you would be heat fixing your sample just like usual. Spread it out a little bit, let it air dry, pass it through the flame to heat fix it. Then after it's cooled down, you can just stain it straight up with like methylene blue for 1 minute, rinse it off and look at it. You don't have to do all these rando steps. So simple staining. Also, one of the things that trip people up on the exam, if I simple stain a gram-positive bacteria and I simple stain that bacterium with, hasn't been stained with anything else. Okay, I want to be clear. If I take Staphylococcus aureus, which is gram-positive, and simple stain it with safranin, what color is it going to be? Red. It's just one stain on it. There's not any other stain there to color it in any other color. So keep that in mind. The most common, I'm not less likely to be too strict, tricky with that and make you think too hard on it. I usually do ask the methylene blue question, which is what I did on the last test. People were still missing it. Don't overthink it. Just because it says that your organism is gram-negative doesn't mean that it's going to stain red. If you're staining it with blue, it's going to stain blue, right? So don't let that trick you up. All righty. Microscopes and their parts real quick. If you observe a specimen at 100x objective through a 10x ocular lens, which is what we have, what is the total magnification? 100 and 10.
Question.
Great. So that is correct. This is going to be 1,000. Good job. Good news. There's not going to be any PCRs, not going to be any DNA stuff on there. That's not going to be on there. Stains and basically your unknown stuff. If it was on the unknown stuff, you have to know it, plus microscopes. So which lens is used with oil immersion? Some people try to do it with all of them. All right, good job. Parts of the microscope, I'm not going to ask you about the nose piece. So FYI.
Thank you.
So other things to know, the parts, what they do would be a good thing to know. Lab safety, things like what do you wear in a lab and all that. So all of the stains, all of the tests, all of the media. I'm really going to be focusing mostly on those stains, the plates and the tubes. There'll be a couple questions on the microscope, maybe one about the lab safety. But just be aware that it's mostly what we've just been doing, and what we did to get to that point. There will be, I'm going to warn you guys because I haven't told enough people about this. You guys remember the checklist that was filled out for our unknown report where you had like marked off positive and negative and what color and whatever that was that you turned in on the first page? Okay, I'm going to have a printed out version, very clearly typed, not any handwriting or anything, that I will have you give you that as well as a correlating dichotomous key. I won't make you memorize that or anything. But you'll have that sheet with the checklist and a dichotomous key, and it'll ask you if you have the gram positive on that sheet, which will be listed. What organism is it? And then you will go through the dichotomous key and figure out which organism it is on that sheet based on what's written out on there. You have everything there. It's just a matter of just following that information on the key and finding it. So it's not going to be remembering, that part should be easy. There's no actually recalling anything. That's just following the answer to the end. But you guys did it for your unknowns. That's how you came to me with your guesses. It's the same thing. It's just all clear and you know the answers are correct. There is a set answer. It's not gonna be weird. Oh, no, it should have made gap, nothing like that. Okay. So yeah, anybody have any questions?
I'm sure I need that luck. Don't even say that.
Of course, you guys can always email me if you do have questions. Okay. Yep. All right.
Thank you.
Yep.
Thank you.
Of course.
The online report was actually really good. I really enjoyed it.
I like it. I like getting to the end where you realize you did it. Like you're like, this wasn't, I didn't mess it up after all.
Working up one day. It was totally worth it.